SANDHAN, All Gujarat Integrated Classroom.
સેટેલાઇટના માધ્યમથી જોડતી કડી એટલે સંધાન.
Good afternoon, students. Today we meet in a new academic year 2012-13 and we continue the program of SANDHAN in microbiology. This program has been well accepted and has been found to be very beneficial to the students. Today, we are going to discuss and talk about a very important topic which is spoken about in the media as well as in the students community of life sciences, and that is the word biotechnology. Basically, at present, students are seeking admission to various courses in life sciences, and one of the subject is biotechnology. So, biotechnology as a subject, what does it offer to the students? What are its scopes? What are the challenges faced by the subject of biotechnology? That is what we are going to discuss today. So, a student should understand that biotechnology as a subject is, as we'll see later on, is an interdisciplinary subject and has a wide number of topics and the subjects to study. It is not a one subject because basic subjects are physics, chemistry, biological sciences, mathematics, etc. But this subject of biotechnology is a gift of this century. The subject is a new one. It is, to be very frank, it is not a subject in itself, rather it's a combination of various different subjects of biological science, that is botany, zoology, parasitology, medical microbiology, human physiology, anatomy, as well as it also includes the other subjects of chemistry, even physics and engineering. So it's a kind of a multidisciplinary subject, and therefore a student has to have the basic knowledge of all other subjects in order to get a proficiency and expertization in biotechnology. So first of all, before I hand over mic to the next speaker, I would like to introduce her. She is Professor Kiran Trivedi, myself Dr. Rakesh Patel. We are both from R.G. Shah Science College, Ahmedabad, and we will be discussing this topic of scope and challenges in biotechnology. Now, what is biotechnology? What are the various definitions? What does the biotechnology mean? That will be discussed by Professor Kiran Trivedi. Madam, please.
Thank you, sir. My dear students, today the topic that we have selected is really very, very interesting, and as the discussion you will come to know that how interesting it is to know something about the biotechnology. As the sir told you that biotechnology is a word you all are very much familiar, but when we want to have something in detail, we should understand that what is biotechnology. So I will first begin with the term, the meaning for biotechnology. Biotechnology, actually the definition for biotechnology has been given by various scientists, various person or the various people who are working in the field, and this definition is according to their own experiences, according to their own experimentations. So if I have to tell you in general, the biotechnology is basically the utilization of biological entities or the cell of higher organisms, may be living or dead, or their components, which like enzymes, which are being used in the process. As a result of that, we get some product or we get some service which can be used in the welfare of human beings. So as a result of this, you will understand that the biotechnology is nothing but it is simply the use of living cells, maybe of higher organism, maybe of lower organism, maybe living, maybe dead, or it can be the use of cell components. Naturally, some of the definition, as I have told you, given by different people. One of them is the definition given by Bullock. Bullock said that controlled and deliberate application of biological agents, living, dead cells or cell components is biotechnology in technically useful operations either of productive manufacture or service operation. Now the point comes, what is biotechnology? So actually, as we understand that biotechnology is not a very modern term, but long back in the year 1919, the Hungarian engineer named Karl-Erecki was the first one to use this term, and he said that any product that has been produced from the raw material with the aid of living organism, we can name that as biotechnology. And according to him, biotechnology means biology plus technology. Technology, as you understand, it is the application of scientific knowledge, skill, material, labor and wealth for practical purposes. Again, if we go for one more definition, it says that any technology that is associated with the manipulation of biological system is biotechnology. In biotechnology, mainly we make the application of scientific and engineering principles in the processing of materials by using the biological agents, so that we get, in turn we get very good service and very good product. Now, initially the production, naturally we may have one question that the production technologies which are related to agriculture, horticulture or animal husbandry may utilize plants and animals. They also generate the useful products, but we cannot call them as, or they cannot be regarded as biotechnology because long recognized and well established disciplines. Now, so what should be called as a biotechnology? Exploitation of animals and plant cell cultures in vitro for product or service as an integral part of biotechnology. So this is how we had come to know about the definition of biotechnology. Now, as sir has already told you that biotechnology is not a single branch, but it is a multidisciplinary science. It combines many more subjects like microbiology, chemistry, biochemistry. On the screen you can see immunology, cell and tissue culture, physiology, downstream processing, biochemical engineering. I mean, so many branches are integrated, and then we have formed this branch or this field as biotechnology. Next, here also you can see the different circles. Each one is representing a different field of biotechnology: agricultural biotechnology, healthcare biotechnology, industrial biotechnology, whatever the word you may use, but they are all integrated with one another. So same is the picture showing all different branches integrated. This is again a same thing shown in the form of a tree, the different branches of biotechnology. Okay. Now, on what principle actually the biotechnological operations they work, or what are the various branches which are being involved? As I have already said that so many branches are being involved in biotechnology. Let us see. When we begin with the operations, suppose if you want to improve some strain, we begin with this improvement of the strain. So improvement of the strain, the procedure will make the use of subjects like microbiology, cell biology, botany, zoology and genetics. Once you have improved the strain, then you have to go for the mass culture that represents the physiology. Then after collection of the mass, you have to continue with the optimization of the microbial responses. Now these microbial responses are processed, or they are associated with the process engineering. Now once you have seen the responses of microorganism or their products, then comes the process operation. Process operation means during which the whole of the yield or the product has to be produced. So that the process operation will involve the various branches like chemistry, biochemistry, chemical engineering and biochemical engineering. So this is how we see that the biotechnological operations are based on different disciplines. Now coming to the next point, stages of biotechnology. Now biotechnology word may be given in 1919, but actually from the very old time, that is in ancient time, the people in the search of various things, they were wandering here and there, and they wanted some shelter, they wanted some food, they wanted so many things. So with the passage of time, they have started growing, and they have started discovering many different things. So ancient biotechnology includes the search of food and use of shelter and domestication of different kinds of animals. Second stage is a conventional biotechnology. Conventional biotechnology means it involves the fermentation promoted for food production and the production of medicine. Third stage is a modern biotechnology. It is a really very interesting one because it involves the manipulation of genetic information in organism, that is what we call as genetic engineering. Now let us see something briefly about the ancient biotechnology. Actually this biotechnology began with the early civilization and developments. There were developments in agriculture and food production. Actually we don't have very firm records about these things, but through the carvings and the sketches and any other archaeological research, we would come to know about this ancient biotechnology. Now coming to the history of biotechnology, you will find that there are many more developments were being made in the year 6000 BC, that is before Christ. Yeast was used to make the beer by Sumerians and Babylonians. Then in the year 4000 BC, Egyptians they used yeast for making the bread and wine. 2000 BC, Egyptians, Sumerians and Chinese they developed the technique for fermentation, brewing and cheese making. 500 BC, Chinese they used moldy soybean curds to treat boils, that is to treat some type of infection. Then Indian ancient literature refers to Sura and Madira, which are basically the fermented products. This is a beautiful picture which shows that in the ancient time how the people used to make the wine by using the wooden big large vessels. Okay, coming to the next stage of biotechnology, conventional biotechnology. But biotechnologists they were using the living organism for manufacturing a variety of useful materials, as for example fermentation of grape juice to produce the wine, antibiotic production by penicillium chrysogenum, microbial production of glycerol by yeast, acetone butanol production by Clostridium acidobutylicum, citric acid production by Aspergillus niger, and production of streptomycin by using Streptomyces graeus. Mutations, recombination and strain selections are the different kinds of techniques which were exploited or which were used to increase the yield of the product. Now let us see something more about the Modern biotechnology, as the name suggests, it is a modern biotechnology, so naturally the techniques which are being used over here are very hi-fi techniques, and absolutely a new product is being obtained by the organisms. Here we are able to engineer a new genetic potential. That means we can absolutely create a new organism, new cell with the new genetic capacity. Technique that has stimulated the progress of modern biotechnology, they includes different one, as for example recombinant DNA technology, gene cloning, PCR technique, plant and animal tissue, protoplast fusion, protein engineering, biosensors, new reactor designs, computer aided bioprocesses. So these are the different modern biotechnological techniques. Now, naturally one of them which is very important in biotechnology is the gene cloning. I'll just give you the short or in brief something about the how the gene cloning is being done. The first step in the gene cloning is the isolation of the target gene from the DNA of an organism. Second step is the insertion of targeted gene into the vector. Vector can be a plasmid or it can be a cosmid. Third stage is the transformation of recombinant DNA in the vector. Fourth stage is the screening of recombinants and selection of transformed cells. Finally, expression of a cloned gene through controlled element is very important. All these steps are dramatically represented in this picture. You can see the picture how the from the bacteria targeted gene has been taken, it is inserted into the vector plasmid, then that plasmid is inserted into another bacterium. Bacterium is a recombinant bacteria, it is permitted to divide, multiply, and so we increases the number of gene, and hence we increases the amount of product, maybe protein or maybe other type of product. So this is the outline of gene cloning procedure. Now the next topic, the scope of biotechnology, will be discussed by Rakesh sir.
Thank you, ma'am. See, madam has already outlined